Excited to announce today one of today's keynote speakers, which is Dr. Paul Turner, who is the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale School of Medicine, and he'll be talking today about phage therapy to combat infections by antibiotic-resistant bacteria. So please join me in welcoming Paul.
All right. Hi, everyone. I hope you can hear me okay. And it's a joy to be with you, at least virtually. It's a great honor to give this talk, and I really greatly appreciate the invitation. Of course, in a perfect world, I would be there in person and be with you. It's not a perfect world right now, so we'll do the best we can. All right, so let me go ahead and share my screen. Hopefully, technology won't let us down. Everybody can see that, I hope. I'll minimize this. All right. So I have a fair bit of ground to cover, but hopefully left enough room at the end for some questions. This is a topic, well, this is a project that's increasingly going on in our laboratory. It really is just one part of our research, but it's going very well, and I'm very happy to share it with you today. What you're seeing here are three pictures that give away a main punchline of what I want to get to. And that is on the left, it's very easy to go out into nature and find bacteriophages, which are viruses that are specific to bacteria. They're incredibly diverse, they're very biodiverse, and they are the most abundant thing on the planet biologically. That's what's estimated. The middle picture is one such of these viruses, a phage that we found in a natural environment. And on the right is the consequence of what we're able to do. And this is a young woman who's in a hospital bed in Texas, and she is undergoing pulmonary decline. And at best, she's going to face a lung transplant. So the same person there on the right, as you saw in the picture on the left, that's Ben Chan, who's a research scientist in my group. And he is showing her a vial of phages that were isolated from the wild, purified in our lab. And she will inhale these over the course of about, I think it was 9 or 10 days, to try to have those phages reach a chronic bacterial infection in her lungs to treat it when antibiotics cannot. So that went well. I'll talk about her case and some of the other ones that we've done. To step back a moment, kind of introducing my broad interest in microbial evolution could be captured in a slide like this. I'm very interested in the genetics, genomics, and evolution of microbes as they face environmental challenges. So this rather busy slide is only kind of showing you different levels of biology where we would expect and where we study viruses and their challenges that they face to infect cells. So starting with the level of molecules and cell biology, we're interested in very proximate protein-protein interactions, for example, that would dictate virus entry or not into cells. And moving into tissue biology in multicellular organisms like us, it is a challenge to viruses to infect cells of different type. For bacteriophages, you could think of this as analogous to infecting different bacterial species or even different genotypes within a bacterial species is not such an easy challenge to overcome. Host defenses, we know increasingly about host defenses in lots of organisms, single-celled organisms like bacteria, and certainly multicellular organisms like us. So we are interested in immunity and other defenses and the abilities and inabilities of viruses to overcome those challenges. And last, we can think about how do viruses move around in the environment through transmission, whether it's via arthropod vectors, direct transmission through sneezing, this kind of thing. So we're interested in transmission and its limitations for viruses to move about in environments. And last would be ecosystem level changes where all biological entities on this planet could be affected by these, including the smallest ones in size, viruses. So we are interested in climate change and global warming and how it would affect all biological systems. To finish talking about my research program, you can think of it as our projects are within these three bins. On the left, I started off working with microbes akin to the way that people use Drosophila or other model systems in the laboratory, and that is just to harness their rigor and power to study general questions in biology, especially evolutionary biology. In the middle of the slide, it's a reminder to say that increasingly we study pathogens themselves, not necessarily model systems, but the pathogens themselves and their ability to evolved to overcome host defenses, for example, and you can sort of put those projects under the realm of ecology and evolution of infectious disease. And on the right is the subject for today, and this is kind of in the realm of evolutionary medicine. Can you take evolution thinking into disease problems and use that insight to improve them? So we're very interested in virotherapy. How can you harness viruses to do good in humans and to solve human problems? So that's what I'll talk about today. And where we will begin is a big public health crisis that is happening. It has not gone away during the current pandemic. As we get that under control, we will increasingly go back to turning our attention a lot to this, the antibiotic resistance crisis, especially considering that some post-COVID survivors, that these individuals might be facing easier ability for bacteria to enter their lungs and cause infections. So the upshot is that wherever in the human body, as well as in other organisms, if you have antibiotic-resistant bacteria overcoming the ability of prior drugs to control them, we have a big problem. And this is indeed projected to be a large problem for humans. To the upper right there, you'll see a projection map for only a couple of decades from now. It is estimated that antibiotic-resistant bacteria could be a leading cause of death in humans and actually exceed the rates of death and mortality caused by cancers. So this is the fact that we're dealing with. An increasing proportion of bacteria, both pathogenic and non-pathogenic, show resistance to commonly used antibiotics. It is a global problem. It threatens lives and economies. The lower left is reminding that big pharma is not really doing a lot to develop new antibiotics. This was a very big target, but now it's seen to produce too little money in return for the investment. And that's because it costs so much money to bring these drugs to market. And largely, if the newest ones that are discovered are placed on a shelf as a last resort drug, then you can understand that a pharmaceutical company can't necessarily keep the doors open if they're not getting the return on the development of this drug and the selling of it. So this has led to what has sometimes been called a discovery void. I want to make sure that I say it's not as if people are not in earnest looking for more antibiotics. I'm just saying that big pharma is showing less interest in this over time. What can one do about this? You can look to an old idea, and that would be phage therapy. Before Fleming accidentally discovered antibiotics, and especially their use took off in Western medicine, before that time, phages had been discovered, and they were even being used in therapy. So let's take a step back and talk about what are these phages. They are viruses that kill only susceptible bacteria. And mostly today, I'm going to talk about lytic phages. That replication cycle in the lower left is a very typical way to show how a phage interacting with a target bacterial cell, if it can bind and have its nucleic acid enter, then it can take over the machinery of that cell, turn it into a virus production factory, and lysis can then occur. So this is an enzyme that is coded by the phage, produced in the bacterial cell, and then ultimately causes that cell to burst open and particles are released. Back in 1917, one of the co-discoverers of phages was Felix Durrell. He only had one academic appointment in his career. It happened to be at Yale University. The point is, in the very earliest days of phage discovery, he was doing phage therapy in animal models and even in people. He was instrumental in the founding of the Eliava Institute. It was eventually named that because the co-founder was George Eliava. And this still exists and operates in Tbilisi, Georgia. So a country that was formerly part of the Soviet Union. If you want to learn more about Felix Durrell and his compelling career, I recommend this book by my emeritus colleague, Bill Summers. So now, phage therapy, even in the earliest days, was seen to hold some benefits. One would be specificity. If you have a drug that's very specific to its target, that's quite attractive because you'll know that it will work on that target. And it has the ability to be a drug that self-amplifies, and that's a very rare occurrence. So in this case, you've got a drug, if you could think of it that way, that makes copies of itself because it's interacting with the cells to do so. And that means you could use minimal dosing. That's what's very attractive about it. Now let's turn to some obvious costs. If you have specificity, it could give you too limited of a spectrum. If you have a drug that is too limited in even the genotypes of bacteria that it can kill in this target pathogen realm, then that's not so good. We'll talk about that more in a bit. The delivery to the infection site could be non-trivial. So it's possible to create a balm or like a salve or something where you put phages in suspension and something like that could be rubbed onto a wound. And that's a long-standing tradition in phage therapy, where this topical use of phages, obviously you're going to be able to directly reach the bacteria that are infecting the body. But other places are not so easy to reach, such as the gastrointestinal system, the bladder, and the lung. So I'll talk about how some sites are easy versus not to reach in terms of bacterial infections when you're using phages. Immune recognition is a big deal. And there are many people who are concerned with the fact that if you use phages more than one time on the same person, the immune system will catch on and see this as a foreign invader and won't allow this to be used as a drug. Anecdotally, we don't see much of that. Interestingly, it could be because essentially humans and most other organisms that evolved on this planet essentially evolved in an ocean of phages, if you will. Today, I guarantee you, you will encounter phages and they will enter your body. You'll drink them in water, you'll eat them in food, you'll sit here and listen to me talk for an hour and you'll breathe them in as they are attached to dust particles and wherever you are sitting. So I'm pretty confident of that. For this reason, humans don't tend to mount a big response to phages, and that's not that surprising. The question is whether that would occur if I administer phages to you to the tune of like 10 to the 13 or 14 particles per day over a 10-day period. But as an evolutionary biologist, what I really thought is a challenge in the modern day for phage therapy is that we have to acknowledge that phage resistance can easily evolve in bacteria. So if you use this therapy, I would predict it will eventually fail because there's so many mechanisms that allow bacteria to overcome phage attack. Some of these mechanisms are summarized here from a recent review paper. To the left, you can think of these as more or less innate mechanisms. So there are, on the exterior of the cell, there could be ways that the cell could change to restrict the ability for the phage to bind or to enter. Once inside of the cell, that nucleic acid could be recognized by restriction enzymes and cut up. So there's lots of ways that bacteria defend themselves immediately and can evolve to improve these ways of defending themselves against phage attack. To the right is a more elaborate system. This is CRISPR-Cas, which I'm sure everybody in the audience has heard of this, but in summary, it's a way for a bacterial population to build up kind of a genetic recognition of common phages that attack that population, and they can immediately deploy defenses against them through these arrays of recognition within the cell at a genetic level. So these are very compelling innate as well as so-called adaptive immunity defenses in bacteria that are able to attack phages that are trying to attack the bacteria. All right, so what are we going to do then? If there are so many mechanisms that bacteria can use to withstand phage attack, this is where something that is commonly seen in evolutionary biology may help. And this is evolutionary trade-offs. So I've studied these in my career, and many evolutionary biologists just generally see this in their work. And what am I talking about here is that evolution involves compromises. So as populations are interacting with some environmental factor to be improved, if possible, through selection and to change through evolution to adapt to these environments, they could do so, but you can't think of this as all traits in these organisms improving at the same time. In fact, sometimes they'll improve in traits by necessity and it'll leave vulnerabilities or liabilities in terms of other traits. So natural selection can improve one trait at the expense of reduced performance in another. There's lots of ways for me to try to describe this. I'm going to stick to human biology for this example. Humans, what do we do better than other animals? Many things. One of them is to hurl objects with high accuracy and speed. And even our closest relatives, the chimps, can't come even very close to what we can do in that realm. On the left there, you're seeing pictures of professional baseball and softball players. They are certainly the best among humans, among the best among humans in doing this. A young chimp there in the middle has a different upper body musculature. And these are our close relatives. They cannot hurl objects with accuracy and speed, but they could hang from a single tree branch, even as a young chimp here, for an extended period of time with their long fingers and their very strong arms. We cannot do that. So these are trade-offs that are seen across species that are close relatives. If you just stick to humans, it's easy to find trade-offs as well. So we are the only bipedal great ape. We're the only great ape that walks around on 2 feet all of the time. So this is wonderful to free up our hands to change the world. Unfortunately, it leaves us vulnerable to lower back pain and to neck pain. So I think that that is evidence of a trade-off even within human biology, that as we've improved through our bipedalism, it has led us to be vulnerable to other maladies. So how are you going to use this knowledge in phage therapy? What we decided to do is update phage therapy in a renewed way to take advantage of trade-offs. So how might this work? Essentially, it's to find lytic phages in the traditional sense, but to find specific ones that are interacting with mechanisms that are responsible in bacteria for virulence factors or for antibiotic resistance. And why is that? The cartoon on the right is illustrating why, and I'll walk you through it. So here's a hypothetical bacterial cell, and I want you to look first at these orange and red protein complexes decorating the cell. These are efflux pumps, and some bacterial pathogens have these. So as an antibiotic enters the cell, it may be pumped out indiscriminately across different antibiotic classes through these efflux pumps. So this is a great way for the bacteria to achieve multi-drug or even pan-drug resistance. If one finds this phage, hypothetically here in red, that is interacting with the outermost protein of an efflux pump for a way to bind and enter that cell, then that bacterial population has a problem. They have to deal with this proximate issue of phages killing cells, and they could evolve resistance to phage attack in many of the ways that I showed you. An easy way would be to change that outermost protein or even to ditch it entirely. So that creates the possibility that you use the phage in the traditional way to kill the bacteria, but you drive the population of remaining bacteria to be enriched for mutants that are now antibiotic sensitive rather than resistant. And this allows us to not only kill the bacteria, but for these phages to synergize with currently approved antibiotics that are useless and to turn them useful again. Here on the right would be a faster example for me to describe. Imagine this purple shield around the bacteria. That could be a capsule that is protecting the bacteria from detection from immune system cells in the human body. So to overcome that, you could find a phage here in purple that is specific to entering only these capsule-forming bacteria, and they will evolve resistance by altering or even ditching the capsule, now making them easily detected by the immune system, changing them from a high virulence to a low virulence state. So what we presumed is that we could find such phages in the wild, that we wouldn't have to necessarily genetically engineer them, but we could take advantage of the billions of years of phage-bacteria interactions on Earth, that if one looked, you could find phages that naturally interact with virulence factors or antibiotic resistance mechanisms. I'm showing here a cartoon from one of our recent review papers that's just highlighting several of these structures, depending on what the bacterial pathogen is, that these could be characterized as virulence factors or contributing to antibiotic resistance. So I'm now going to walk you through a pipeline of discovery that we've used effectively to find such phages and to use them in people. The first is to remind that finding phages in the wild is pretty easy business to do. You can look in pristine environments like that lake in the upper left here in Connecticut, or you could look in heavily altered environments where humans have really heavily impacted them, such as the shoreline of Haiti and this river in the Democratic Republic of Congo. Those happen to be endemic cholera regions. So if one wanted to find phages against cholera-causing cholera disease bacteria, then you might look there. On the right, not so glamorous, but a very easy place to find phages would be like a sewage treatment plant. And the lower right would be an agricultural farm, where we know increasingly that the use of antibiotics in animal feed, this is preferred by the farmers because it increases the mass of the animals, but it also can enrich for antibiotic-resistant bacteria in that geographic location. So we've gone to farms often to look for phages that are specific to those bacteria. Once we find them, we bring them into the laboratory and we subject them to whole genome sequencing. I'm a big proponent of this. The price is always coming down, and I think immediately this gives you a lot of useful information. You can take your phage candidates and place them into different virus families. So what I'm showing you here is a recently acquired about 100-member phages that were taken from the wild, and we fully sequenced them. We know that they're lytic phages that are specific to Pseudomonas aeruginosa, an opportunistic bacterial pathogen. And this gives us this immediate information. It also lets us find any evidence that some phages that we could use in therapy we would worry if they have, say, toxin genes or other things that would be dangerous potentially to the patient. Sometimes those genes are carried around by phages. So seeing homology to known dangerous genes would cause us to remove those phages from our candidate libraries. Moving forward in the pipeline, one would want to know for a candidate phage, how good is it at killing genotypes of the target bacteria? I said earlier that the specificity of phages could be a downside to using them in therapy. So some of this unpublished work, again by Ben Chan and my group, would be if you take these phages here on the left that happen to all belong to this one virus family, and you challenge them in this case to infect clinical isolates of Pseudomonas aeruginosa that have come from infected people. And what you'll see in this matrix is that everywhere there is a square in dark blue, that's where the phage on the left was capable of successfully infecting that challenge bacterium on top. We can do whole genome sequencing on the phages as well as the bacteria and try and use that information to help us understand the patterns in this matrix. And let me just say at the outset, there's often not an easy pattern that emerges here. It's quite complicated, and there could be many reasons that the bacteria are or are not susceptible to phage attack. But the good news is that I'm showing you already that some viruses, including certain phages, are naturally broad in their host range. So you're seeing a lot of dark blue in this matrix, and that suggests that these phages that have very likely never seen those bacteria are immediately capable of killing them. And if they're very broad in their killing ability, they could be attractive for use in therapy. Now I'll step away from this large data set and reveal the size of it. And I know it's too small for you to see the details, but it's mostly to impress upon you that some of these data sets are very large. And to make sense of them as a human through ordinary analysis is pretty difficult. So we've taken machine learning and artificial intelligence tools to sort through the data to try and make an accurate prediction that if I just knew the genome sequence, say, of a bacterium, could I predict which phages are able to attack it from my library? So I'm excited that this kind of work is happening in microbiology. I'm also excited to hear more about the machine learning that Dr. Davis will talk about this afternoon at the symposium. But this is the direction that this kind of research is going. All right, so the next step in the pipeline would be if you find a phage that is broadly capable of killing bacteria, what I talked about earlier is for us, the best candidates would be if it is binding to an antibiotic resistance or a virulence factor mechanism. So some of the work of my graduate student, Katie Courtwright, was published last year looking at a technology that gives you a high throughput platform to try and discover cellular receptors that phages use. And maybe I'll just remind that in virology, this is a very difficult task. We've heard a lot about ACE2 protein and binding of SARS coronavirus 2, certainly in the last year. But for the vast majority of viruses, it's kind of tricky to figure out what they are using on the cell surface as binding. For example, HIV is certainly among one of the best studied biological systems ever in science. And there still remains some mysteries, the total number of cell types in the human body that HIV is able to infect. All right, so I digress for a moment. I'll come back to this InSeq platform for high throughput discovery of cellular receptors in the phage bacteria systems. How would this work? So let's take, for example, if you did this in E. coli, you could use a transposon mutant library where the transposon is coming in and disrupting individual gene function. And then you subject that to phage selection for some phage of interest. And what grows out would be enrichment for the phage-resistant mutants. In other words, if the gene is disrupted that is important for the phage to use, especially if it's something that's presented on the cell surface, if it's not there, then those bacteria that have undergone disruption of that gene will be favored and they'll be overly abundant in the output. And the output looks like the lower left. So we are going to be looking in a moment at data sets that were subjected to deep sequencing of the prior library as well as the output after the selection. And we're interested here in what's showing up in the upper left in blue. Those would be genes that are for phage infection. So if you now look at these two examples from her paper, in the middle, if you take a phage that is well known and it infects E. coli, and the literature is telling us that a long time ago, it was determined that for phage T6, that it is able to infect and kill E. coli by interacting with the TSX nucleoside porin. Well, what she's finding in her analysis in this control is that that is the highest statistically significant result in the output, and that's quite satisfying to see. Here on the right would be a phage example where this is a phage that's lytic, it infects E. coli, and that's essentially all we knew about it before we put it into this analysis. And it also, through this process, shows us that it is using the TSX porin as binding an entry. Now, don't think that this is the only receptor that coli phages can use. It just happens to be the same one here, and I'm showing it for a reason that will become obvious later in the talk. But in short, this is a way to figure out how to infer what phages are binding to on the cell surface and to do some follow-on experiments to confirm that this is the case. So next, taking all that in mind, let's look at this figure, a cartoon from a recent paper that I published with another postdoc in my group, Alita Burmeister. And what I'm emphasizing is that if you know through this pipeline, you can find phages that are broadly capable of killing your target bacteria, and they're interacting with a virulence factor or an antibiotic resistance mechanism, we think that this could benefit phage therapy. So in this cartoon from our review paper, I basically walked you through these first two examples. If you have a phage interacting with efflux pumps, if you have a phage interacting with a capsule around the bacterium, you can get the benefit of the evolution of phage resistance, in this case leading to antibiotic sensitivity there for the efflux pumps. And additionally, if the bacteria become resistant to phages and change that capsule, it can also allow easier entry of antibiotics. Here on the right is one that I didn't say before. I just want to highlight it, that if you find a phage that is interacting with LPS, lipopolysaccharide that surrounds the exterior of the cell, then you can think of this as like a forest of trees growing up from the cell surface, and it has a particular canopy height. And a lot of phages come in and they'll go to the mid part of that canopy to infect the bacteria by attaching to LPS. Now, what will happen next is that the bacteria die, or you're going to get enrichment for mutants that have kind of shaved down or reduced the LPS. It's become more shallow. And that presents a problem for the bacterial population, because with shallow LPS, there's easier entry of antibiotics. And it's yet one more mechanism that we can select for antibiotic sensitive bacteria using a phage. So some quick evidence of this from recently published work, sticking with E. coli as a model. Consider here on the left the Toll-C efflux pump, which is very well characterized in the system. This is not my work here, this very beautiful structure, but it's mostly a reminder for me to say that in this case, the outermost protein is called Toll-C. Now, a phage that we discovered in nature, U136B, we found that it selects against antibiotic resistance in these bacteria. First of all, look here in the middle. If you take several E. coli strains that have engineered for their individual gene to be knocked out, this is a way of depicting how well does this phage grow on these different challenged bacteria. And you'll find that if you knock out certain genes, the phage is just fine. However, if you knock out Toll-C, its growth is below the limit of detection. And that's what we would like to see in terms of, aha, then it means Toll-C is essential for this phage to enter and kill the cell. Here on the right is the phenotypic consequence. Starting first, in the absence of any antibiotic, the wild type E. coli, this Toll-C knockout engineered mutant, and a spontaneous phage-resistant mutant of E. coli all grow normally in an antibiotic-free environment. If you place tetracycline into that environment, as expected, the Toll-C knockout cannot grow very well, and indeed the phage-resistant mutant can't grow that well either. And that's through data I'm not showing you right here. It's because it underwent a mutation in Toll-C to resist phage attack, and the consequences, it made it antibiotic sensitive for tetracycline. Now, the plot thickens for this phage because it's interacting with LPS as well, and I'll quickly walk you through that. As we showed in this paper last year, here's a side view of LPS, and I'm color coding the portion of LPS where we know now where this phage is coming in and using it as a co-receptor. So it's not only binding to Toll-C, it is also using a portion of LPS as a co-receptor. Here in the middle, you can find that if you knock out certain genes that contribute to LPS formation, knocking out those here in the middle of the forest canopy are creating a problem for this phage, and it cannot grow when certain genes are knocked out because the LPS portion is missing. And the phenotypic consequence here is on the right. For these phage-resistant mutants, they can overcome phage attack, but they've now altered their LPS, and it leaves them more vulnerable to a different antibiotic called colistin. They are now more sensitive to colistin than are the wild type bacteria. All right, now I've given you a lot of information, but I'm going to take it a little further to say that I'm emphasizing how trade-offs would help phage therapy. So if we could deploy that phage I just showed you against E. coli, and it's killing the bacteria as well as enriching for TOL-C mutants or LPS mutants that are antibiotic sensitive, that's wonderful. However, I can't control the spontaneous nature of mutations in the target bacteria. So we examine this in this paper, where we would be especially worried if there's a tendency to see what are called trade-ups. Suppose that you get the evolution of phage resistance, and instead the bacteria become more resistant to antibiotics rather than sensitive to them. So how do you examine that? You can take 20 mutants obtained at random that are allowing these bacteria to escape the phage attack by changing TOL-C, and then you could measure how sensitive or resistant are those bacteria to tetracycline. And here you see the ancestral level, or the minimum inhibitory concentration of tetracycline that kills the wild type bacteria. And this bar here, oh, this is good news. Now we've got 6 spontaneous phage-resistant mutants, and they show this decreased ability to grow in the presence of tetracycline. This bar is not so great news. So not only can phage resistance occur for the E. coli to keep the ancestral level of antibiotic resistance, but also you can get instances of increased tetracycline resistance. Here on the right is a bit better news overall. When we find these spontaneous LPS mutants, largely they either have the ancestral level of resistance to colistin, or they are worse at growing in the presence of colistin. So the main point in showing you this is that there are a variety of ways that the bacteria can mutate to avoid phage attack. Many of them lead to a trade-off that we would like to see, but admittedly some of them lead to a trade-up or to a non-change in the resistance to the antibiotic. So now imagine I'm deploying this in a person. I can't control any of this, and I'm telling you that there are different mutations that could lead to phage resistance. Something else becomes important. How competitive are those phage-resistant mutants with one another to determine which one or ones would come to dominate that population after the phage selection has occurred? So we looked at this in the laboratory by looking at the cost, the growth cost of the acquisition of phage resistance through these 2 pathways, either TOL-C change or LPS change. So here, if you look at the output over 24 hours, the growth of the bacteria is inferred by change in optical density. The black line is the growth of the wild type bacteria. Those depicted in purple are the TOL-C mutants, and they are growing just fine, thank you very much. Having acquired phage resistance, they don't seem to be showing any cost of that relative to the growth of the bacteria in a benign environment. Here in green, the LPS mutants have a big problem. They seem to be showing pretty low fitness compared to the wild type, as well as the TOL-C mutants. So if I just let this occur, where if you co-evolve the bacteria and if you take the wild type bacteria and co-evolve it with the phage, my prediction would be that the TOL-C mutants are going to win out, because spontaneous mutation can happen anywhere to solve this problem. But I'm showing you that the TOL-C mutants are not suffering a big fitness consequence. So that's essentially what we observed. And this is only a snapshot of the data from the paper, but focus first here on these lineages in red. That's just when you evolve the bacteria on their own through experimental evolution. And if you track their phenotypic change in tetracycline resistance, you don't get any change. And that doesn't surprise me, because there's no tetracycline in the environment and there's no phage either. In bluish green here, if the phage is present and you let coevolution occur, then any mutations can pop up, and it becomes a matter of which of these mutants are competitively superior. You would see a trending downward in tetracycline resistance closer to sensitivity at the end of this experiment if the TOL-C mutants are winning the competition. So I'm not showing you the genetic data. That's what we observed, but the phenotypic consequence is that on average, we're seeing tetracycline resistance wane in these populations, because the TOL-C changes are the way that the bacteria is solving the phage problem. So 2 more slides before I get to some clinical evidence. I've talked a lot about antibiotic resistance switching, and I want to give a little airtime to virulence switching through our approach. So I'll introduce now another phage taken straight from the wild, TIFPH6, and it selects against virulence in Pseudomonas aeruginosa. So it is given this name TIFP because we found that it is able to bind to the type 4 pilus of Pseudomonas aeruginosa. Here on the left, similar to the data I showed you before from that 2020 paper, but these are unpublished data where for this phage, if we put it through that in-seq screen, we find that there are actually these highly significant hits for a variety of genes that are contributing to type 4 pilus biogenesis in this system. So knocking out those genes allows the bacteria to escape phage attack, and that's because the phage is having to use the type 4 pilus to bind and enter the cell. But this has a cool consequence for virulence, and that's because there's a gene network in these bacteria that links up type 4 pilus production with the production of a virulence factor called piocyanin. And this has a very toxic effect in the human body. It causes cellular inflammation and oxidative stress. So what we found is that if you have these bacteria losing their pili to overcome the phage problem, this would affect their biofilm formation because the pilus is important in that. It affects their mobility because twitching motility occurs with the type 4 pilus, but it also interferes with this piocyanin production, like I said. Here on the left is a test tube where we've pelleted the wild type bacteria, and you can see that this blue-green color of the supernatant, that's because they produce piocyanin, and it has that color and they're making it at a high abundance. But here on the right is a phage-resistant mutant that is now completely debilitated for the production of piocyanin. So here are some unpublished data that focus first on the bars in blue. If you take Pseudomonas aeruginosa and expose it to various antibiotics, you can find that some of them have no effect in killing the bacteria, and unfortunately, it'll cause them to produce more piocyanin than they ordinarily would, and that is an obviously bad thing. Erythromycin is compelling because it's an antibiotic that is given to people to calm down the cellular inflammation that results from piocyanin production, but it's not killing the bacteria necessarily. It is just reducing the piocyanin production in a way. Here on the right in red, these are data for this phage, and we find that it does both. So here on the far right, this phage is not only killing the bacteria, but the end result is that it is greatly reducing piocyanin production in the bacterial population because the mutants that are left over are debilitated for that. And it works very well in the presence of erythromycin, as well as Cipro, which had this deleterious effect on the left. And this heat inactivated control says that if you just kill the virus particles, then you don't get that effect. So these unpublished data are really compelling because it says that you should be able to use this phage in people that are infected with Pseudomonas aeruginosa, killing those opportunistic pathogens and reducing the very debilitating effects of the piocyanin production in any bacteria that are left over on average. All right, so in my remaining time, I'm going to turn to clinical evidence. One of the things that surprised me the most from entering into this kind of research was the immediate ability to use it in people. And I admit I was kind of naive. So I entered into phage therapy research, always interested in it, knowing that evolution of phage resistance was a problem and could we overcome it. But I underestimated, even here in the New Haven area alone, there are so many people who are already on the front lines of the antibiotic resistance crisis and may need emergency help because antibiotics are not killing their infections. So let's look at some of the evidence that we've amassed here locally as well as elsewhere to use these phages to help people out. So we have what could be called a pond to bedside pipeline in my lab. And here are the same pictures I started with, highlighting that we've got phage discovery, characterization, and ultimately treatment. Others are trying to do this, a handful of other laboratories, but increasingly there are phage centers across the United States trying to develop this technology. There was this nice book written about a famous case at UC San Diego where phages were used to help a man who was dying of drug-resistant sepsis, bacterial infection, and phages were used to overcome that and to save his life. So this has a lot of promise. Our first case is a published one, so I won't talk about it in detail, but it had the story of a man who had undergone an aortic arch replacement, which is not a trivial surgery, but it actually happens quite often. And it's an example of how you can take an artificial thing and put it in the human body safely. But the problem is it becomes a substrate for which an opportunistic bacterial infection can take hold on that substrate and become chronic. And if it is antibiotic resistant, you've got a big problem because you may not be able to remove it. This man, by the time his physicians found out that this was the case, with Pseudomonas aeruginosa infecting him, that was multi-drug resistant, the infection was so bad it had eroded away his sternum. So it was quite remarkable that this man was even alive. And he came to our attention and we talked with his head physician, a surgeon, and immediately this man and that surgeon bought into the idea of phage therapy because there weren't any other options that they could give him. They could try the surgery over, but he was expected to be too elderly and frail to survive a second surgery. So the long story short is that I wouldn't be talking about this unless it worked. And here we are in a picture, Ben Chan and I in this man's office after we were able to administer this phage therapy. It worked against his infection by administering a phage and a useless antibiotic that synergized because this phage was causing the antibiotic to be useful. And this worked. We were thanking him in his office. I don't know if you can make it out. He has a thank you card and a Phage Plus toy. So this was covered in the news quite a bit. It was also mentioned in a Scientific American article a couple of years ago. But I want to say here, full transparency, is phage therapy here to stay? Well, that's an open question in the Western world. I would say in Eastern Bloc nations like Poland and places like Russia, phage therapy never went away. They invested more there in phage therapy than we did, whereas we invested a lot in antibiotics, which are currently failing. So the main idea here is whether phage therapy will really take off in the West and become part of more of traditional approaches. So the young woman who I started with for those pictures at the beginning, let's look at her unpublished case a little bit closer. So in her case, she has cystic fibrosis. So this is a genetic disease that causes the lungs of these individuals to be having widened airways, overproduction of mucus, and sort of other features that allow bacteria to come in more easily and cause chronic infections. And certainly Pseudomonas aeruginosa, this is happening in such patients, and it's increasingly of alarm because these bacteria are becoming multi-drug and pan-drug resistant, and they are everywhere. Pseudomonas aeruginosa is a very common bacterium. So here on the left, when this young woman's case came to our attention, she's only 22 years old, and she's suffering with a chronic P. aeruginosa infection for which her lung function was extremely poor, her exercise performance was poor, and the estimated amount of these bacteria in her lungs was at the highest level measurable. It's called a +4, as evidenced by her sputum that she was able to cough up, and that was analyzed. But worse, she was showing an infection that was pan-drug resistant. So all of these antibiotics listed here, her infection was showing evidence of being resistant to all of them. So essentially, there were no other options from an antibiotic standpoint for this young woman. And you can look at these many years of decline in her lung function. This does happen in some CF patients, such that by the time she reached our attention and asking for help, she really needed it. Otherwise, she was going to undergo a lung transplant at best, if not just face pulmonary failure and death. So we administered this therapy by putting phage into her nebulizer, and she inhaled it in the hospital 3 times in one day, did not show any adverse consequences of that, was sent home to finish the nebulizer administered phage therapy over the course of 9 more days. And all throughout that time, her lung function was being tracked. And you could see that after this therapy, her lung function jumped up. And as we tracked her further forward in time, it continued to be stable at a better level than this decline that was observed. But best was this. So not only did her function of her lungs go up, avoiding the necessity for a transplant, some of the bacteria were left over. That's not surprising because it's very difficult to remove bacteria that have invaded the CF lung. But those that remained were showing the evidence that we'd seen in vitro that this phage therapy could reverse the antibiotic resistance and make it to antibiotic sensitivity. So now this woman and her physician has a lot of options in case these bacteria outgrow to cause what's called an exacerbation. And now they could deploy these antibiotics seemingly to control the problem. So her case, I went through it very briefly, and this is a rather busy table. The first 2 rows in it are the 2 individuals I just talked about. The other ones, I don't have time to present their cases and yet more, which would fill a larger table. This is actually a typo. We've treated over 20 chronic infections, most of them pulmonary, to date with 0 safety concerns. And Often they've been CF patients, as you've seen in this table, but those who have non-CF bronchiectasis or chronic obstructive pulmonary disease, there are very many more people with those problems than there are individuals who are born with CF. So we've been happy to help some of those individuals as well, and they certainly are being targeted for us for help in the near future. So some more clinical evidence from these patients. We've treated so many of them now that we're trying to bundle them together in what's called a case series. And here are some of the data from the unpublished case series. I said that these patients who are treated for pulmonary problems, they inhale the phage over time, but they can also cough up sputum that we can analyze. So looking first at lung function as well as bacterial density in the sputum, they go in the correct direction. And these are statistically significant results when you bundle cases together and look at them in the same analysis. Lung function improvement goes up. Bacterial load, as evidenced by the bacteria numbers in the sputum, go down for the bacterium we're trying to remove. Here on the lower right, for those patients where we expect to see reversion of the antibiotic resistance changing to sensitivity, we've often observed that. Now here is an inflammatory molecule reduction outcome that is highly significant, and let me show you some more details of that. So in using, say, that type 4 pilus targeting phage that I talked about earlier, in collaboration with our physician partners, John Koff and Gail Stanley, we've taken patient sputum longitudinally over time, and we've analyzed it to see in this example, do you have clones coming from the patient's sputum that you analyze in the laboratory and find some evidence that the piocyanin production is actually going down? And that's what this graph on the left is showing you. So several clones taken on day 1 and day 5, etc., are showing this pattern of eventually troughing out so that the piocyanin production is not even measurable anymore. And that's shown here on the right in a very obvious color change, that if these early clones are analyzed and they're making blue-green supernatant, those later in phage treatment are not. That's because they're debilitated in piocyanin production. So what does this have to do with inflammation? I mentioned that earlier, that this virulence factor contributes a lot to cellular inflammation. So if you look at these clones taken from patients, spin them down in the laboratory, remove the cells, but you take the supernatant, you could put that on airway epithelial cells of human origin in tissue culture. And you could examine how stressed out are those human cells by the supernatant that you're presenting putting onto the monolayer. And you can measure things like IL-8 and IL-6 and find that there's a statistically significant drop in the inflammation response of these human cells when you're using supernatants from late in the phage therapy, from clones that were taken late in the phage therapy. And that's very satisfying because that's the direction we predicted. All right, so to finish up, how well is in vitro prediction working in patients? So this is definitely a black box, and I'm as shocked as anybody that it's working pretty well. Now, I'm going to relate this briefly to some of what we're suffering through now in the pandemic and the research that goes on in my laboratory that I haven't talked about today. And we're trying to better predict the evolution of virus emergence. That's a big part of my research program. So what we would like to know is in the future to be able to better predict coronavirus or HIV-like or even influenza virus pandemics by knowing more about these systems and predicting when these could be consequential. So are generalist viruses more likely to emerge on hosts such as humans? This is one of the predictions that we've made, and that generally people think maybe this is the problem. As viruses evolve to be very generalized, and they're very good at jumping onto hosts that are new to them, such as humans. Well, if you take that question and think about it in a different way, we're trying to find phages that are very good at broadly killing target bacteria. And we would like to know, why are some phages good at doing this? But we also don't want them to be too good, right? We don't want them to kill so indiscriminately that they'll easily jump onto bacteria, say, of the microbiome and emerge on them. So I think that these questions of how do viruses emerge to cause disease, as well as how do you use virotherapy in a safe way so that the virus doesn't sort of emerge onto cells that you don't want it to evolve to use. So we take these questions to be hand in hand, and we're writing a review paper on this currently. So how phage therapy affects many things in the human body, we simply don't know, because we're deploying it as fast as we can to help safely through personalized medicine in human patients, but we are not so naive to think that other things could be going on. Interactions with your cells, it is possible for phages to enter human cells, but not replicate in them. And I'll show you a little preliminary data on that. Interactions with phages in your viromes of your microbiomes is a possibility. Changes in bacterial traits that we are not predicting is something that we might worry about. Interactions with non-target bacterial pathogens, as well as other pathogens that are not even bacteria, as well as any effects with the resident microbiome. So a couple of tidbits here would be interactions with human cells. In collaboration with Jen Bomberger's group at University of Pittsburgh, one of her postdocs, Paula Zamora, has looked at the ability of some of our phage therapy candidate phages and those that have been put into people. They can enter human cells and even elicit an interferon response without replicating in human cells. So this is important to know, is how much interaction is directly occurring between phages that are deployed in therapy. Even though they don't seem to be harming human cells, there is an interaction. Here on the right, I'll just quickly say that some published work talks about what are called jumbo phages, those with large genome sizes that can sometimes enter cells here, bacterial cells, and replicate, but they'll shield off their replication from detection by DNA CRISPRs. And we had actually deployed a phage like this without fully understanding that it has this side benefit. So the expectation is this phage is more easily capable of avoiding CRISPR defenses when put into humans to solve a bacterial infection problem. And that's a side benefit. So one thing that I could just sort of say at a 30,000 foot view level is that doing this work, as I've talked about it today, kind of sets aside what are the interactions that could be going on across the microbiome, across a community of either pathogens or non-pathogens. So one of my students, Mike Blazinin, he has a paper that's coming out soon in ISME Journal that appreciates through a meta-analysis what is the evidence in the literature that if you look at phage bacteria interactions through ecology or evolution, in the presence and absence of a community, does it make a difference? So we can take published data from other people's work, and in this meta-analysis, we're showing examples like this one, that if you examine the interaction between Klebsiella bacteria and a phage that infects it, over time, if they're just together in the same flask or some environment, they could coexist. But if you put them in the presence of a speciose community, here adding only 2 other members of different species, you find that there's a big difference. And in this case, Klebsiella is now outcompeted, or it disappears from the environment when these other community members are present. So the pipeline that we're trying to build is already versatile, but it's growing. I've talked a lot about P. aeruginosa today, especially in the patient treatment. But you can think of this as trains running down the track in parallel, and some of the most advanced ones, we're already using them preclinically in personalized medicine, but we want to move them to this final column here of clinical trials. And that's what we have as a next step. There's a clinical trial now underway at our hospital, abbreviated here as Sci-Fi, but we are looking at the ability of phages to protect the lungs of healthy volunteers who have cystic fibrosis. And the idea is, over a multi-month period, can we find that those people who receive the phage are better protected than those who only receive a placebo. We teach this in courses at Yale. So we have a course-based undergraduate research happening in the first year for Yale students who want to take this course to learn about this technology. And lastly, we've taught it in workshops around the world. So in places like East and West Africa, where they might want to rely on this technology soon. And last, I'll say that non-human systems suffer through antibiotic resistance as well. Plant and animal agriculture and aquaculture, as well as zoo and aquaria animals, can be suffering with these infections. And we've tried to look at these systems, and also immediately tried to help a sea turtle that was suffering with a multi-drug resistant bacterial infection in Florida, and that worked out beautifully. And last, I'll say that the next steps, one often asks me this question: if you deploy phages worldwide, would you just kind of recreate the problem of the antibiotic resistance crisis, where now you'll suddenly have phage resistant bacteria as problematic and they're overcoming phage therapy? And I would say no. We talked about this in a paper last year. If you'd like to go check it out, it's an opinion paper. And that's because horizontal gene transfer is really contributing to multi-drug resistance spread in bacteria, especially conjugative plasmids. But that's not how phage resistance typically could spread, even if it does spread in bacteria. And lastly, broad spectrum antibiotics would exert general selection on bacterial species, both pathogens and non-pathogens that encounter it, to evolve resistance to antibiotics. That'll never happen, in my opinion, with phages, because even the most generalized phages, at best, you're trying to cover the spectrum of genotypes in the target bacteria, and you're not going to be killing indiscriminately bacteria that are outside of that species. So I'll end with thanking the many people who contributed to this work. I know I've kind of run a little bit long in this, so I'm going to quickly just thank my current and former lab groups. I mentioned many of the individuals here on the right, but I'll end by saying Deepak Narayan, he was the surgeon who helped us with that first case, and he believed in what we were doing and allowed us to help more people. But without his initial belief in what we were doing, that never would have happened. We unfortunately lost him to cancer in 2018, but he was, I always like to think of him and thank him at the end of my talks because he really opened the door for this project to go forward. So I will stop there and happy to take questions if you think we have any time.